The most important skull in early human origins is not the most complete, the most famous, or the easiest to explain. It is a broken face and brain case from the eastern shore of Lake Turkana, catalogued as KNM-ER 1470, and usually placed under the name Homo rudolfensis. At first glance, it looks like another fragment in the crowded story of early humans in Africa, another fossil from the Great Rift Valley puzzle, another specimen placed between Australopithecus and later humans. But the deeper problem is that this skull sits in the center of a contradiction that has never fully gone away. If KNM-ER 1470 is a separate species, then early human evolution in Africa was a branching thicket of small populations, each with different faces, teeth, brains, and bodies. If it is instead a large male version of Homo habilis, then the whole structure begins to shift. The small faces and small bodies, long treated as typical habilis, become one side of a sexually dimorphic species, and the larger skulls and skeletons, long treated as something nearer Ergaster, become the other side of that same biological range. In that reading, the familiar story of Homo erectus beginning in Africa becomes much less secure. The skull was found in 1972 near Kubifora in northern Kenya, in deposits near 2 million years old. From the beginning, it created tension. Its brain was far larger than the tiny Australopithecine skulls that had shaped earlier arguments, yet its face was wide, flat and strange. It had a broad mid-face, a long palate, large tooth sockets, and a facial architecture that looked neither fully like habilis nor like classic Asian erectus. The cranial capacity was often placed around 750 cubic centimeters, a brain size larger than many habilis fossils and larger than some Dmanisi skulls from Georgia. But the face was not sleek, narrow, or unmistakably modern in its proportions. It was a mosaic. That word is often brought into early human evolution when a fossil refuses to sit obediently inside a neat sequence, and KNM-ER 1470 is exactly that kind of fossil. It looks advanced in one direction and primitive in another, and the more we compare it with other early fossils, the less comfortable the traditional categories become. The classic interpretation gave KNM-ER 1470 its own species, rudolfensis. That solved one problem by creating another. It allowed scientists to keep smaller fossils such as KNM-ER 1813 in habilis, while placing the larger, flatter-faced 1470 somewhere else. On paper, this looked tidy. In practice, it created a crowded landscape in which several human species all occupied eastern Africa at roughly the same time, all with overlapping brain sizes, overlapping teeth, overlapping tools, overlapping habitats, and frustratingly incomplete skeletons. The picture became more complicated than the fossils could really carry. If each difference in skull size, face width, tooth size, and body size receives a species name, then early human evolution becomes a taxonomic maze. But if those differences reflect sex, age, body size, and variation within one strongly dimorphic species, then the maze begins to flatten. This is the radical interpretation. Rudolfensis is not a separate branch at all. It is a large male habilis. In that model, the smaller skulls that have shaped the mental picture of habilis are not the whole species. They are the smaller side of it, likely including females and smaller males. The larger skulls are not a different kind of human. They are the other side of the same species. This kind of size gap is not strange in primates. Gorillas show immense male and female size differences. Orangutans show extreme contrasts between large flanged males and smaller females. Even some extinct Australopithecines show heavy sexual dimorphism, with males much larger than females. The question is not whether such a pattern can exist. The question is whether early habilis had that kind of body plan, and whether later scientists broke that variation into too many species because they expected early humans to fit a cleaner ladder. The implications are enormous, because habilis has always been a difficult ancestor for Erectus. It has a smaller brain, a more primitive face, a different body plan, and in many interpretations a more ape-like set of proportions than later humans. Yet the earliest African fossils assigned to Ergaster, often treated as African Erectus, arrive with bodies that look taller, longer-legged, and more efficient for travel. The traditional story turns that into progress. Habilis gives way to Ergaster, and Ergaster becomes the African beginning of Erectus. But this story depends on reading the fossil sample in a very particular way. It depends on treating size and robustness as evolutionary advancement instead of variation inside one species. It depends on seeing the smaller habilis specimens as primitive ancestors and the larger Ergaster specimens as a new grade of human. If those larger fossils are simply large-bodied habilis, then the clean transition breaks. That is why KNM-ER 1470 matters so much. It sits exactly where the categories are weakest. It is too large-brained for the simplest picture of habilis, yet too primitive in its face for a straightforward Erectus ancestry. It forces a question that is rarely asked plainly enough. What if early humanity is not a sequence of sharply divided species, but a wide biological range that has been split again and again because the sample is fragmentary? In a living population, skulls vary, males and females vary, young and old individuals vary. Geography creates local patterns, nutrition changes body size. And when the fossil sample is tiny, every large male has the potential to become a new species, while every small female becomes evidence of another. The danger is obvious. We can mistake a family portrait for a species tree. Dmanisi in Georgia sharpened this problem. The site has skulls and jaws from around 1.8 million years ago, and they come from a group of individuals buried in the same general setting. They show a surprising range of shape and size. Some have tiny brains near the lower end expected for early humanity, while others are larger. Some faces are more projecting, some jaws more massive, some skulls more lightly built. If those fossils had been found scattered across Africa, each one in a different valley, each one without the others beside it, there is a strong chance that they would have been sorted into different species. Instead, Dmanisi forces us to confront variation inside a real population. It shows that one early human group could contain skull differences that look, under the older African system, like several species. That is the point at which Dmanisi becomes more than a Eurasian site. It becomes a test of the African taxonomy itself. If Dmanisi individuals with different skull forms, different brain sizes and different jaws belong to one population, then why should the African fossils automatically be split into habilis, rudolfensis, Ergaster, and African Erectus? The usual answer is that Africa has more time depth and wider geography, so the variation has greater meaning. That is fair as far as it goes, but it still leaves the same core problem unresolved. The African fossils are often too fragmentary to carry the weight placed on them. Skulls lack bodies, bodies lack skulls. Jaws drift from one category to another. Isolated teeth are asked to carry species labels. The result is an elegant chart built on bones that rarely connect. The Dmanisi fossils also challenge the idea that the first humans outside Africa were true Erectus in the classic Asian sense. Their brains are too small, their faces too primitive, their bodies too modest. They look far more like a mobile, variable early human population than like the fully developed form known from Java and China. That matters because Dmanisi sits outside Africa very early. If the humans at Dmanisi were already outside Africa around 1.8 million years ago, and if they resemble a habilis grade more than classic Asian Erectus, then the first expansion out of Africa was not carried by the tall, familiar textbook Erectus. It was carried by something smaller, more primitive, and more variable. In plain terms, the first thing out of Africa was closer to habilis. This changes the direction of the story. The old model starts with Africa as the birthplace of Erectus, followed by expansion into Asia. But if early travellers were habilis grade, and if classic Erectus appears more clearly in Asia than in Africa, then a different possibility opens. Erectus, in the stricter sense, formed in Asia after an earlier habilis grade population left Africa. In that scenario, Africa did not send out fully formed Erectus. Africa sent out an earlier population, still primitive in brain size, face form and body variation. Asia then became the region where the classic Erectus pattern took shape. Thicker cranial bones, long, low skulls, strong brow ridges, a more consistent cranial architecture, and eventually a wide spread across Eastern Asia. This is where the African evidence becomes thinner than the textbook language suggests. The phrase African Erectus sounds solid, but the actual skulls are not as clear as the label implies. Olduvai hominid 9 is often brought into the conversation because it has a large cranial capacity and a heavy brow. It is an impressive skull cap, but it is not a complete face, and brain shape studies have complicated its placement. If its internal brain case pattern sits nearer habilis than classic Erectus, then size alone cannot settle the issue. A large brain case is not enough, robustness is not enough, a thick brow is not enough. Without the full package of classic Asian Erectus traits, Olduvai hominid 9 becomes something more ambiguous, perhaps a large African early humans, perhaps a late robust habilis-grade individual, but not a simple proof that Erectus flourished in Africa. Dhaka from Ethiopia has also been placed in the African Erectus or Ergaster discussion, but it sits later and belongs in a different conversation for many researchers. Its age and anatomy bring it nearer the broad Middle Pleistocene pattern often associated with Heidelbergensis. That does not make Dhaka irrelevant. It makes it dangerous as evidence for the origin of Erectus. A later African skull with more derived traits cannot easily prove that classic Erectus began in Africa hundreds of thousands of years earlier. It can show that African humans evolved in their own direction. It can show that mid-Pleistocene Africans had robust, large-brained skulls. But that is not the same thing as proving that the original Erectus package was African. The southern African child skull, often brought into this debate, adds yet another layer of difficulty. A toddler skull is not a stable foundation for a species claim because young individuals have faces and vaults that change dramatically during growth. Juvenile fossils are valuable, but they are also hazardous. The features that define adult identity are not fully formed. Brain case shape, brow structure, facial projection, dental development, and cranial thickness all shift during maturation. Calling such a skull Erectus with confidence stretches the evidence far beyond its strength. A child can tell us that an early human population existed in southern Africa, but it cannot carry the whole burden of proving classic Erectus in Africa. The deeper issue is that Ergaster itself has become a bridge term, a way to keep the African story stable while admitting that African fossils are not identical to Asian Erectus. In many textbooks, Ergaster is the African version, taller, more modern in body, less specialized in skull shape, and ancestral to later humans. But if the large African fossils are not a clean species, and if the smaller fossils are not a different species, then Ergaster begins to look less like a true biological group and more like a sorting label for large early human bones. The famous long-limbed body from Nariokotomi, often called Turkana boy, has shaped this view more than any other fossil. Yet Turkana boy is a juvenile. His adult size is reconstructed, not observed. His skull and body are remarkable, but one skeleton cannot define an entire continent-wide species. Turkana boy also creates a contrast that has been too easily explained away. If he stands for Ergaster, then Ergaster looks tall, long-legged, and almost modern below the neck. But if other habilis-grade fossils were much smaller, then scientists have to decide whether that contrast is species difference, sex difference, nutrition, age, local ecology, or some combination. The usual answer has been species difference. The alternative is that early humans in Africa was far more dimorphic than later humans, with large males and smaller females creating the illusion of multiple species. In that model, Turkana boy and the larger skulls are not the beginning of Erectus in Africa. They are the upper end of a broad habilis-grade body range. This also explains why the face of KNM-ER 1470 is so disruptive. It is large, but not Erectus-like. It is robust, but not a classic Asian Erectus skull. It has a broad face that looks almost too primitive for the role it has been given. If it is male habilis, then the problem vanishes in one direction and deepens in another. It vanishes because 1470 fits as a large male inside a variable early African species. It deepens because the boundary between habilis and Ergaster becomes unstable. Once the large male side of habilis is recognized, some fossils placed nearer Ergaster stop looking like a new species and start looking like the same animal scaled upward. This is not wild speculation. It is part of a long argument about how to interpret early variation. Some researchers split the fossils into many species. Others see fewer species with wider ranges of variation. The splitters emphasize differences in face shape, tooth size, cranial capacity, and jaw form. The lumpers emphasize overlap, dimorphism, small fossil samples, and the cautionary lesson of Dmanisi. The debate persists because the evidence is real on both sides. KNM-ER 1470 really is different from smaller habilis skulls. Dmanisi really shows wide variation inside one population. African early fossils really are fragmentary. Asian Erectus really has a clearer classic pattern than the African material. The issue is not whether differences exist. The issue is what those differences mean. The strongest version of the African Erectus model depends on a chain of assumptions. It assumes that larger African early fossils represent a new species, rather than large individuals. It assumes that Erectus is a coherent African ancestor rather than a grade. It assumes that Dmanisi is an early Erectus population rather than habilis grade migrants. It assumes that later African skulls can illuminate the origin of a much earlier Asian pattern. Each assumption is possible. Taken together, they create a smooth story, but the fossils themselves keep interrupting that story. The small-brained Dmanisi skulls interrupt it. The broad-faced KNM-ER 1470 interrupts it. The ambiguous Olduvai hominid 9 brain case interrupts it. The later position of darker interrupts it. The child skulls interrupt it. Every time the story becomes clean, a fossil makes it messy again. A different reading is simpler in some ways. Early habilis in Africa was highly variable and strongly sexually dimorphic. Large males and smaller females were later split into different species names. A habilis grade population left Africa before the classic Erectus form existed. At Dmanisi, we catch one branch of that early expansion in real time, small-brained, variable, adaptable, and already outside Africa. In Asia, some descendants developed into classic Erectus, with the long, low skull, thick cranial walls, strong brow ridges, and more consistent anatomy recognized from Java and China. Africa, meanwhile, continued along its own path, producing later robust humans that ultimately connect to Heidelbergensis and other Middle Pleistocene forms. In this version, Erectus is not an African invention. It is an Asian development from an earlier African dispersal. That interpretation changes how we look at the whole early human story. It strips away the comforting ladder from habilis to Erectus to Erectus and replaces it with movement, dimorphism, and regional transformation. It also forces humility. We are not looking at a complete archive of early humans. We are looking at scattered fossils, often broken, often isolated, often named before enough comparative material existed. A skull found without a body becomes a species. A jaw found without a skull becomes a mystery. A juvenile becomes a landmark. A large male becomes a new branch. Then, years later, another site like Dmanisi arrives and shows that early humans varied more inside one population than many models allowed. The real mystery of rudolfensis is not simply what name belongs on KNM-ER 1470. The real mystery is why this skull still holds so much power over the origin story of Erectus. If 1470 is its own species, then Africa was crowded with several human forms, and Erectus still needs a clean ancestor. If 1470 is male habilis, then habilis was larger, stranger, and more variable than expected, and Erectus loses much of its foundation. If Dmanisi belongs with that same habilis grade expansion, then the first humans outside Africa were not the classic long-headed Erectus of Asia, but a more primitive traveller that carried an older African body plan into new landscapes. And if classic Erectus truly crystallized in Asia, then one of the most familiar claims in human evolution has been standing on a far less secure base than we were taught. Thank you for watching. The question is not only where Erectus began. The deeper question is whether we have been looking at early African fossils through categories too narrow for the animals themselves. KNM-ER 1470 still stares back from the edge of Lake Turkana with a face too broad, a brain too large, and an identity too unstable to ignore. It can be called rudolfensis, or it can be seen as a large male habilis, but either way, it exposes the same problem. The origin of Erectus is not a straight African staircase. It is a broken trail of fossils, names and assumptions, and one skull from Kenya still has the power to turn the whole map around.